because of only 2 lectures, so my intention is we give you a very superficial understanding of what pharmacokinetics and pharmacodynamics are. Feel free to provide feedback to Chris, Kalila, or me as to whether the pitch is right, whether something is too superficial, too much, and it's hard to digest, then I will modify for subsequent years. So my 2 lectures are split. The first one, we're going to look at pharmacokinetics. And we'll see how much time is left. And it may need to be got into second lecture as well, because even though you look at 24 slides, but I can promise you, I can easily spend 45 minutes on 20 slides. So 24 is like a record for me. Okay, so you will see for my lectures, you always see a list of lecture learning outcomes at the very beginning. So it gives you an idea of what I intend you to learn from this particular lecture. And when it comes to assessment or exam and so on, hopefully you get an understanding of, okay, how much, to what extent you are expected to learn or remember. That's the purpose of these learning outcomes. And let me know if they are not clear or not specific enough. I'm more than happy to revise for subsequent years or clarify for this year. Now, so before I touch on pharmacokinetics, I will just briefly touch on the concept of drugs. So you will see on the screen, there are certain words that are grayed out because we are going to look at the other half in my second lectures. But for this first lecture, we are going to look at the concept of drugs. So drugs, well, according to the most recent definition, is a substance that can produce a biological effect when it's introduced to our body. Now you appreciate that most drugs that we administer to patients, they are used to treat symptoms of a condition rather than a cause of a condition. But there are exceptions. So you are doing antimicrobial drugs. These are the exceptions because they are used to kill the pathogens that cause those conditions or infections, for example. Now, also important for you to know is that most drugs are used for treatment, but there are drugs that can be used for prevention. So one of my favorite topics that I would like to talk about, not necessarily in these lectures, is anti-HIV drugs, because we do use drugs not only for treating HIV infection, but also use some drugs for HIV prevention. Okay, so because you are doing a unit in Master of Infectious Disease, so if I use examples in my lectures, I will try my very best to look at the drugs, the antimicrobial drugs that are currently used, so that you can see the relevance. Now, in terms of drugs, they are broadly divided into 2 groups. They are small molecules. They still represent the majority of drugs that are being used today, but progressively we are looking at biologics. They are large molecules, vaccines, antibodies, MRs, and so on. So these, because the molecular size, they can't be administered orally, just like other small molecules, we can take it orally. But large molecules, they can't. Why is that? Because they cannot be absorbed. So we are going to touch on this in a few slides. So there's just 2 examples. Tenofovir disoproxil is the example I use quite frequently in my 2 lectures. It is a drug that is used for HIV treatment and prevention. Now, what is unique about this drug, which I will touch on later, is that it's actually a prodrug. So what is a prodrug? I will explain to you later, but this is an example of a small molecule. On the other hand, infilviatide, this is a peptide consisting of 36 amino acids. Again, this is also used as an anti-HIV drug, but because it's so large, it cannot be administered orally. It must be administered intravenously. So it will touch on how different drugs are administered by different routes. We are not going to look at drug targets in this lecture, but we'll touch on that in my second lecture. Now, the second slide, we're going to look at what is pharmacokinetics and pharmacodynamics. Now, I'll begin by looking at a layman's language. So let's consider a drug and a human body. So pharmacology is really to look at the interactions between a human body and the drug. So because we are concerning human pharmacology, it's not animal pharmacology, so we're going to look at drugs and the interaction between the drug and human body. Now, when we talk about pharmacokinetics, we are talking about what the body does to a drug. So once a drug is administered, or for example, taken orally or administered intravenously, how our body processes that drug. We are going to look at different 4 processes later. So that's pharmacokinetics. The other side, pharmacodynamics, asks the question, what a drug does to our body. So how a drug, for example, if someone has a headache, they may take paracetamol, and how paracetamol works in our body. So drugs don't work magically somehow once it's in our body. You need to get to the right site and interact with the right molecular target and initiate maybe relevant pathways in order to produce the therapeutic effects. So that's something you need to keep in mind. So that's pharmacodynamics on the top, which will be covered in my second lecture. Now going back to pharmacokinetics, we are going to look at 4 important processes using the acronym ADME, ADME. So it represents absorption, distribution, metabolism, and excretion. Now if you look at the right-hand side, there's a diagram nicely summarizes what each of the 4 processes represent and what it means using a layman's language. So I'm going to take you through each of the 4 processes in subsequent slides. You'll see a word transporter in the center of the diagram. And that's because transporter is important for each of the 4 processes. So I won't touch on the role of transporter for each of those, but I will show a few examples to illustrate that when we look at drug design and the action of drugs, why it's important to look at whether transporters can play a role in how the drug behaves in our body. Now we are going to begin by looking at the first process, absorption. Now you will see the definition is provided at the top of the slide. So for that reason, I don't intend to repeat what is already written on the slide. Now when you look at the movement of drugs, I would like you to consider our human body are compartmentalized. For example, if the drug moves from the intestinal lumen across epithelial cells and into our bloodstream, they need to cross layers of cell membranes, and therefore layers, cell membranes, serve as the boundary of different compartments. So a drug, if they move to one compartment to another, they need to cross cell membranes, which are essentially lipid membranes. So that's very important for you to keep in mind. Later, when we look at different processes, particular distribution and excretion later, the same principle applies. The drugs need to move in order to undergo those processes. Now you will see, I hope I have a stick somewhere so that I can point out stuff. But you can see here, maybe use my hands, how drugs may move across the lipid cell membrane depending on the physical chemical property of the drug. So by the way, when I talk about these processes, I am focusing on small molecule drugs, not biologics, because they have a different set of rules, because they are large molecular size. Now for some water-soluble drugs, they can cross the cell membrane using aqueous channels. But for drugs that are lipophilic, then they can cross the cell membrane through the lipid bilayer. Some drugs can be transported to the other side of the cell membrane using transporters. So that's the first time I talk about transporters, like carrier proteins. Now we talk about different routes of drug administration. And as far as antimicrobial drugs are concerned, they are either administered orally or intravenously. And these are the 2 most important routes of drug administration. And as far as possible, I will only focus on drugs that are administered through these routes, unless otherwise specified. So that gives you an idea of how drug may be absorbed. Now, I mentioned that it's important to consider the physical chemical properties of drugs. So I'm going to take you through some important properties and how we can use experimental approaches to examine the properties of different drugs. Now the size does matter in terms of drugs because as a general rule, the larger the molecular size of the drug, the less likely the drug will be absorbed. All those drugs will be absorbed poorly. The other thing is how the solubility of the drug. Bear in mind that when a drug moves from one body compartment to another, they need to be able to navigate both aqueous environment as well as lipid membranes. So for drugs, they need to show a bit of hydrophilicity, but also a bit of lipophilicity in order to survive those two environments. Now, for those drugs that are hydrophilic, then the absorption, if they are administered orally, then the absorption can be poor. Now, in that case, prodrug can be a useful strategy to improve oral absorption of those drugs because by introducing a prodrug, it increases the lipophilicity of the drug, and therefore they are more likely to be able to cross the cell membrane and get into our systemic circulation. And we're going to touch on the concept of prodrug later. Now this shows three examples as to how the solubility of drug can determine how a drug moves. For a hydrophilic drug, you will see that it hardly gets through the lipid cell membrane because of a lack of lipophilicity. For drugs that are reasonably lipophilic, they can move across the cell membrane quite easily. But for drugs that are very lipophilic, they can move across the cell membrane, but they may get stuck there. So they end up staying in the lipid membrane rather than going to the other side of the cell membrane. So for the drug that is capable of navigating different environments, it's best to have a bit of hydrophilicity, but at the same time some sort of lipophilicity. So both rather than two extreme. Otherwise, they will either not get through the lipid membrane or they get stuck in the lipid membrane. In both cases, they are not desirable. Then the third physicochemical property of a drug concerns whether the drug is charged. And for most drugs, they are classified either weak acids or weak bases. So depending on the pH environment where the drug is in, these drug molecules will be ionized to different extent. Now just bear in mind that if a molecule carries a charge, it will not be able to cross the cell membrane because cell membrane is lipid, and they will get stuck in the environment where they are highly ionized. So for some drugs, this can be useful clinically. For example, if you want to, and one example I can come up with is aspirin poisoning. The one way is to increase the pH of urine so that the drug molecule is highly ionized, and therefore they cannot be reabsorbed back to our systemic circulation, and therefore the drugs will move from our body faster, and that's a way to treat aspirin poisoning or overdose. But that's an example, clinical application of the relevance of whether a drug is ionized or not. Now, in terms of how we can assess the solubility of a drug, there are two methods. First one is known as log P. P means partition coefficient. The way to do it is put a drug into two layers of solvent. One is organic, using ethanol, for example. The other one is water. So you mix the drug with those two layers of solvents vigorously and wait until it settles, and you can measure the concentration of the drug in both layers. So you will appreciate for a drug that is lipophilic, then you will see a higher concentration in the layer of ethanol, the organic layer of solvent. But if a drug that is hydrophilic, then you will see a higher concentration in the water layer. But that will give you an idea of whether the drug is lipophilic or hydrophilic or both. The other way to look at the solubility of a drug is to measure the polar surface area. Now, historically, this was done experimentally, so it requires the determination of the 3D structure of the molecule, and then identify different atoms, for example, nitrogen and oxygen, and the hydrogen attached, and then you can then work out the poly surface area. But there's also a computational method known as topological polar surface area. So what it happened is that there's a table that shows different fragments and the corresponding PSA value. For a molecule, you just calculate how many these fragments a molecule contains and look it up in a table and then add them up. That will give you a fairly accurate PSA. So without going through all these steps on the right hand side. So you can see these two. There's a very strong correlation between topological PSA and experimentally determined PSA. So these are the two ways by which you can actually use to determine or assess the solubility of the drug, and then determine whether you need to modify the structure so as to improve oral absorption. How are we traveling? Am I speaking too fast or not clear? Good? Okay, I can see thumbs up. Okay, now, this example we're going to look at, again, my favorite drug, tenofovir. So the one in the middle is actually, well, I would say the two drugs on both ends, they are both prodrugs. These prodrugs have little or no pharmacological activities. So they need to be converted to an active drug in our body to display therapeutic effects. Now, what's unique about this drug is the one in the middle, tenofovir itself, is not yet an active drug because the drug needs to be phosphorylated twice so that it can be incorporated into the elongating DNA sequence to inhibit the synthesis of DNA. But I won't go into that into much detail. Now, so the reason why we use these two prodrugs is that tenofovir itself is hydrophilic, so the oral absorption is quite poor. By adding an ester group to the drug to form either tenofovir alafenamide or tenofovir disoproxil, these drugs are more lipophilic, and therefore the absorption is better because all these drugs are administered orally. Now, these two drugs, even though they are both prodrugs, they are converted to tenofovir at different locations in our body. It turns out how the drug is converted in our body is important because for tenofovir disoproxil is converted in the plasma. So this actually is linked to the toxicity of the drug because the whole body is now exposed to tenofovir. On the other hand, tenofovir alafenamide is only converted in T cells, and therefore that is the site of action, right? So the drug targets HIV, and HIV infects CD4 positive T cells. So it makes sense for the drug to be converted inside the cells in those targets rather than in the plasma, because then the whole body will be exposed to the drug, and that is linked to toxicity. So in that sense, not only how well the drug is absorbed matters, also how these prodrugs are converted in our body can determine whether a drug will cause toxicity or not. So this is a good example to look at these two drugs, and where they are converted to tenofovir determines whether the drug causes toxicity or not. Okay, that is drug absorption. What's the time? Let me keep an eye, because I can talk a lot without realizing how much left in my lecture. I think that is the end of drug absorption, the first process associated with pharmacokinetics. Oh, one more thing, okay. So remember I talked about the importance of transporter. Now for a drug that is administered orally, it will need to cross the epithelial cells from the intestinal lumen into the systemic circulation. Now the problem is that the gut wall express transporters that for some drugs, they are able to return the drug to the intestinal lumen. So in terms of drug absorption, that is not desirable. Okay, so for a company to design drugs, so the FDA has actually developed a guide to the industry that as a part of preclinical studies, when they look at these in vitro experiments, they should look at whether an investigation drug is likely to be subject to transporter-mediated efflux, which means that it returns the drug to the intestinal lumen. So CACO2 cells is an important in vitro cell model for this purpose. The reason is because they express important efflux transporters, as you can see here. So it's a good model to see whether the drug will be subject to efflux mediated by these transporters, and then that may require modification of drug so that it's not susceptible to efflux or not considered drug at all. So this just shows the setup of the CACO2 cells, and the drug can be added either to the apical site or the basolateral site, and then you can measure the concentration on the other side. And then I won't go into the detail as to how the assay works and also how you can interpret the results, but just give you an idea of this is a model that you can use for in vitro studies to assess the drug movement. Now I really got to speed up because I still have 3 processes to go through. So the second part, we can look at drug distribution. So again, the definition is provided at the top. Now it goes back to the point I mentioned earlier that the body is compartmentalized. So for a drug to move from one compartment to another, they need to cross cell membranes and layers of cell membranes. Now this shows a few examples of how drugs may be distributed into different body compartments. Some drugs may simply stay in the systemic circulation without being distributed into tissues, but some may be distributed into interstitial fluids but not getting into cells. But some drugs may be highly accumulated into tissues because of their physicochemical properties or because they have strong tissue binding characteristics. So I'm going to take you through some of those factors that can influence how a drug may be distributed into different body compartments. Now pH, so I briefly touched on pH earlier using the case of treating aspirin poisoning. That is an example of how changing the pH value of one body compartment in order to change the ionization of the molecule, and this will lead to ion trapping. Now blood flow. Different organs receive different amount of cardiac output. So for organs or tissues that are well perfused, then drugs are likely to be distributed into those organs first. So for example, the lungs, the liver, the heart, and so on. And in resting conditions, skeletal muscles and fat, they are poorly perfused, and therefore drug distribution into these organs or tissues will be poor compared to other well-perfused organs. Now, tissue binding. I talked about drugs can potentially accumulate in certain tissue and organs. Now, one example I would like to mention here is tetracyclines. So these drugs bind calcium with high affinity, and therefore they tend to accumulate in bones and teeth. Now, for this reason, tetracyclines are classified as category D drugs by the TGA, which means that they should be avoided in pregnant women or young children, because otherwise, if they are given to pregnant women, then these drugs can affect the growth of bones in the developing fetus, and the newborn may end up with malformation. So tetracyclines is actually classified as human teratogen, which means that it can cause irreversible birth defects, so in particular in terms of bone growth. And so that's an example of how drugs may accumulate in tissues or organs because of their binding probability. Now, plasma protein binding, I won't touch on too much, but you see that there's proteins in the circulation where drugs can bind, and these will affect how much free drug molecules are available. And one point I want to comment is that only free drug molecules can get to the site of action to produce therapeutic effects. If the molecules are plasma protein bound, then they cannot produce those actions. So we talk about the solubility of drug, and the same applies here. And permeability of blood tissue barrier. That's probably more important if the drug will need to get to the brain to act. For example, you want to treat an infection in the brain. Then blood-brain barrier serves as a barrier to remove drug or return drug to the systemic circulation or to the capillaries, because the mechanism is there to protect our brain. So I won't go into much detail in that unless you're dealing with drugs that need to act in the brain. Then blood-brain barrier would be something you need to consider, because normally drugs are very difficult to penetrate into the brain because of the presence of the barrier, unless there's some inflammation that will move that barrier. Now, I'm glad that we are getting to drug metabolism, because I'm going to spend quite a few slides on this process and talk about many interesting scenarios and why it's important for us to consider in terms of drug development. Now, drug metabolism allows the body to chemically modify drugs so that to avoid potential toxicity caused by exposure to toxins or xenobiotics or harmful chemicals. And liver is the primary site or primary organ for drug metabolism, because the liver expresses a variety of metabolizing enzymes. And there are other organs or tissues that are also capable of metabolizing drugs, but they play a minor role in most cases, depending on the drug. But the liver is the most important organ in terms of drug metabolism. Now, for a drug that is administered orally, then one thing for us to consider is that whether the drug will undergo first pass metabolism, that which occurs in the gut wall and the liver. So you will see here for a drug that is, should I actually use the mouse here, if you can see the movement of mouse? Or should I use my hand? So you see for a drug that is administered orally, this is the small intestine, they need to cross the gut wall. First, they get to the portal vein. And portal vein will take the drug to the liver. And only those drug molecules that survive the gut wall and the liver will end up in the systemic circulation. And that is the portion of drug that is bioavailable. So if a drug undergoes extensive first pass metabolism in the gut wall and the liver, then the bioavailability of the drug is going to be low, and that's something we need to consider when we design a drug. We don't want that to happen, otherwise you need to increase the dose of drug, or the drug is not going to be good at all. Here's something you need to consider. Now also you see that in the intestine and the livers, it shows a range of enzymes that are capable of metabolizing drugs. And I will talk about one particular family later, but just highlight that the range of drug enzymes that are involved in drug metabolism. Now, in the next few slides, we're going to look at the outcome of drug metabolism. The first is to increase the molecular size and hydrophilicity of the parent drug. The reason is that this will then facilitate excretion of the drug. Remember, for a drug, if they are hydrophilic, then they are less likely to cross the cell membrane, and therefore they are less likely to be distributed into different organs or tissues. Then they will be easily removed, or more easily removed in urines. Now this diagram shows the fate of a drug depending on whether it's lipophilic or hydrophilic. So for drugs that are lipophilic, they typically will undergo metabolism in the liver first, so that they become more hydrophilic before they are excreted into the urine. And for drugs that are itself hydrophilic, then they typically don't undergo metabolism in the liver. They can be excreted into the urine straight away. And so for this reason, you have to consider whether a drug is hepatically metabolized or simply renally excreted, because that's important when in terms not so much about drug development, but also when we look at how the dose of the drug, then we need to consider whether we need to adjust the dose depending on whether the drug can be affected by impaired liver function or impaired kidney function. Now, the other outcome of drug metabolism is to decrease the half-life of a drug. So half-life basically tells you the time it takes for the plasma concentration of the drug, for example, to drop by half. So on the left-hand side, without drug metabolism, you can see it takes longer for the concentration of the drug to drop by half, but in the presence of a drug metabolism, the half-life shortens. So that's another outcome of drug metabolism. Now the third outcome of drug metabolism is that it can change the pharmacological activity of a drug, or it can potentially introduce a toxicity. The most common outcome, though, is that the parent drug is pharmacologically active. The drug produces therapeutic effects, but once the drug undergoes drug metabolism, then the metabolite is inactive, and then the metabolite will be removed from body through excretion and elimination. So that's a very common outcome. There are other possible outcomes. The drug itself is pharmacologically active, and the metabolite itself is also pharmacologically active. Then we'll extend or prolong the action of the drug because both the parent drug and the metabolite are active. So that's fine. Okay, that's not an undesirable outcome. The third one, a pharmacologically active parent drug is converted to a toxic and reactive metabolite. One example I use in many of my lectures is paracetamol. Well, when we use the recommended dose, paracetamol is safe. The problem with paracetamol is that in the event of overdose, it's converted to a toxic metabolite that cannot be detoxified quickly enough by our body. So it ends up with an accumulation of the metabolite, and that's where the metabolite causes damage to the liver, and in severe case, the outcome will be death. So you look at news from time to time, you'll find articles talking about someone died of paracetamol overdose because of severe damage to the liver. That's one possible outcome here. The last one, we talk about prodrug, so which has little or no pharmacological activity, and it needs to be converted to a metabolite that is pharmacologically active. So I go back to the example of tenofovir I mentioned earlier, but just keep in mind that even though the two prodrugs are converted to tenofovir, tenofovir itself is not yet a pharmacologically active drug because it requires the next two steps to be phosphorylated twice before it becomes pharmacologically active. Okay, what's the time? Let me keep an eye. 32, okay, seems that my pace is reasonable. Now, I'm going to talk about one important drug metabolizing enzymes and in vitro method for us to investigate whether a drug undergoes cytochrome P450 mediated metabolism. So this is important. I won't go into the classification and so on, or the nomenclature, CYP3A4 for example, but I just want to highlight some of the important CYP enzymes. The first one is CYP3A4. It has a very broad range of substrate specificity, and it metabolizes a whole range of drugs. And for this reason, this enzyme is often involved in drug-drug interactions. So what do drug-drug interaction mean? I'm going to give you 2 examples later, but just keep that in mind that because of CYP3A4 is has a wide range of substrate specificity, and therefore it's quite susceptible to CYP-mediated drug-drug interactions. Now there were others important ones. 2D6. 2D6 is more important in the context of single nucleotide polymorphism, but you can see some others, but I don't expect you to remember any of these. I don't think the learning outcomes require you to do that. Now I'm going to look at the right-hand side. So it's an in vitro assay to look at whether a drug undergoes CYP-mediated metabolism. So this is preclinical study in vitro. The way to do it is collect the livers from rats or mouse and then smash it so you can produce homogenates, and then the homogenates will then undergo centrifugation a few times at ultra high speeds, and each centrifugation will separate a certain components, and at the end you will end up with this microsomal fraction where the microsomes are rich in CYP enzymes. Then you can use these microsomes to conduct in vitro study to look at whether a drug undergoes CYP-mediated metabolism. Now we talk about drug metabolism and also mention the word or the phrase drug-drug interactions. So what does it mean? So that occurs in the event of co-administration of multiple drugs, or in some case drug and food, but I'm going to focus on co-administration of multiple drugs. There are 2 scenarios, but the most common one is CYP induction. So before that, I'll briefly touch on most common one is CYP inhibition, but I will begin by looking at CYP induction. So certain drugs has the ability to increase the expression of certain CYP enzymes. What it means is that if another drug is metabolized by these particular CYP enzymes, that means the metabolism will be enhanced because of increased expression of that particular enzyme. We are going to look at one example later to iterate CYP induction as well as CYP inhibition. Now CYP inhibition is more common. That occurs when 2 drugs bind to the same enzyme. These 2 drugs don't necessarily need to be the substrate for the same enzyme. One can be a substrate, the other one can simply bind to the same enzyme. Now this diagram briefly illustrates what happens. So in the absence or just in the presence of one single drug, there's no competition with another drug, so the drug is metabolized normally. But in the presence of another drug that they both bind to the same enzyme, there will be a competition. Now what happens is that the metabolism of one drug or both drugs can be reduced, and this may end up having a very high or higher than expected concentration of both drugs, and that can potentially lead to undesirable effects depending on how much the concentration has risen. So there are 2 possible outcomes. Either you observe exaggerated drug effects because of a reduced drug metabolism, or in the event of a prodrug, then you won't see any effect at all because the drug is unable to be converted to an active metabolite. Now I'm going to show you an example, and again, apart from midazolam, which you don't need to know, the other 2 drugs are antimicrobial drugs. So let's first look at midazolam alone. So the curve shown by the unfilled circles. So that's what happens. It shows the plasma concentration time curve. So it shows you the plasma concentration of the drug over the course of time. And this is the one with midazolam. Now just keep in mind that the drug is metabolized by, well, you don't need to remember the CYP3A4. This is just an example to illustrate the concept of CYP induction and CYP inhibition. Now rifampicin is a CYP3A4 inducer, so you remember that inducer increases the expression of CYP, and therefore you will appreciate that the metabolism will be enhanced, and therefore the plasma concentration of the parent drug midazolam should decrease. Make sense? Increase metabolism, and therefore the parent drug will be reduced in terms of plasma concentration. That is what you, hang on, oh yes, here. The unfilled circle and the filled circles. That's what happens when you co-administer with inducer. The metabolism is increased, and therefore the plasma concentration of the parent drug dramatically reduced. Okay, this is an example of CYP induction. Now, it's conazole, so it's an antifungal drug, so it's a CYP3A4 inhibitor. Now, based on your understanding, it will inhibit the metabolism of midazolam, and therefore you will expect that the plasma concentration of the drug will be increased. That's exactly what you see here. If you look at the filled triangle and the unfilled triangles, that's because the drug is unable to be metabolized, and therefore the plasma concentration is higher than if the drug is alone. So this graph nicely shows the concept of CYP induction and CYP inhibition. Now we talk about CYP inhibition. It's very common. It's actually clinically beneficial. So again, I'm going to show you two examples. They are both used as part of anti-HIV drug treatment. The first one, ritonavir itself, is a drug, anti-HIV drug, but the clinical use is not with that property. It's actually used as a CYP3A4 inhibitor, so that when another protease inhibitor is used in combination with ritonavir, then ritonavir is used to inhibit CYP3A4, which normally would metabolize the other protease inhibitor. So this one is to inhibit the metabolism of another drug. So the reason is that so that you don't have to use, you can use a lower dose of the other drug in the presence of a CYP inhibitor. The other one is cobicistat. Again, it serves as a CYP3A4 inhibitor, but then it's used in combination with some other drugs. So the effective drug is highlighted, abitacavir. You don't need to know what the drug does, but I can tell you it's an integrase strand transfer inhibitor. Again, is to inhibit the metabolism of this target drug by CYP3A4. So these are examples of drugs where drug-drug interactions is actually clinically beneficial. And these drugs are known as pharmacokinetic enhancers because they are used to enhance the pharmacokinetic properties of the other drugs. Let's quickly look at this example. So this is saquanavir alone. You can again see the plasma concentration here, but when it's combined with ritonavir, you can see a dramatic increase in the plasma concentration of the target drug because, again, the metabolism of the drug is inhibited, and therefore you can see an elevated plasma concentration. Okay, one last slide on drug metabolism. Let me check the time. Okay, I think I'm still on the right pace. So drug metabolism is probably the most important process out of the four. I'm not saying that the other three are not important, but if you ask which one is the most important out of these four, then drug metabolism will be the one. That's because when you look at these enzymes, not every individual expresses the same level of the same enzyme. That's because single nucleotide polymorphism or even polymorphism will determine the expression level of a particular enzyme, and therefore the activity of that particular enzyme. So you find that drug metabolism varies between individuals because of their genetic makeups. And so in that kind of sense, single nucleotide polymorphism is particularly important in a clinical sense. Now you can see another pie chart. It's different from the one you saw before. This shows the number of SNPs identified for some of the important cytochrome P450 enzymes. The most important one is 2D6 because it has a whole large number of single nucleotide polymorphism. And so on the right hand side, this chart shows 4 groups of the population can be broadly divided into 4 groups depending on the number of copies of CYP2D6 genes. So it can be ultra-rapid metabolizer, extensive, intermediate, and poor. And you can see, depending on the number of copies of that gene, the plasma concentration time curve varies between these 4 groups of individuals. It can range, the outcome can be ranged no effect because the concentration is too low. By the way, I will come back to a plasma concentration time curve at the very end of the next lecture once we have looked at PD and PK. But on the other hand, the concentration may be too high that the drug not only produces therapeutic effects but also toxic effects. And one example here, which I didn't include, is the conversion from codeine to morphine. That's a very typical example to illustrate this. But otherwise, this shows the impact of single nucleotide polymorphism. Will the recording stop after? Will you continue? Okay, but I'm sure all of us deserve a break. So this is the last slide of drug metabolism, is to look at the importance of single nucleotide polymorphism, to look at pharmacogenetics. So the combination of pharmacology and genetics. So actually it's quite an important topic now, because when we talk about dosing or prescription of drug, sometimes it's important to look at the genetics of the individual because it will determine whether the drug is appropriate or not, or whether the dose needs to be reduced or increased, or whether the drug should be ditched at all and switched to another drug. So that's quite important to look at the genetics, the importance of genetics in pharmacology. Now the last bit, drug excretion, I don't think there are many slides. Again, you will see the definition is provided at the top of the slide. Now on the left-hand side, it shows a number of routes of renal excretion. The most common one is through urine, so via kidneys, and therefore kidney is the most important organ in terms of drug excretion. And because of this, the kidneys are susceptible to drug-induced toxicities. There are some examples associated with antimicrobial drugs. And the liver, on the other hand, is also susceptible to drug-induced toxicity because it's the main organ for drug metabolism. So liver and kidneys are 2 organs that are susceptible to drug-induced toxicity. Now, I think you remember this same diagram I showed earlier is to look at the fate of a drug depending on whether it's lipophilic or hydrophilic. So I don't intend to repeat the same information again. Now, just this slide is really to show you the 3 important steps involved in renal excretion of the drug. The first step is glomerular filtration. So it talks about how the drug, small molecules, are filtered through glomerular capillaries to form filtrate here, the first step. And just bear in mind that this process is age dependent. As we age, our ability to the glomerular filtration will drop, and this can also be affected by diseases. So that's something particularly important if a drug undergoes renal excretion. The second step, reabsorption. So just bear in mind that as the filtrate goes through the tubules, water will be reabsorbed back to the capillaries, and therefore that will increase the concentration of the drug. And potentially that will lead to a concentration gradient that allows the drugs to be reabsorbed back into the capillaries through a passive diffusion. And depending on the urine pH, the drug may undergo different degree of ionization. And just keep in mind that I mentioned earlier, ionized drug molecules, they are unable to diffuse freely across cell membranes because they are charged. And so this can be used clinically to modify the degree of drug ionization, and therefore they will change whether the drug is reabsorbed or not. So again, goes back to the example I used earlier in terms of aspirin poisoning. Now, the last step, active secretion. So it occurs primarily in proximal tubules, and this is mediated by transporters. So this is the second time I mentioned the role of transporters, and transporters is important for all 4 pharmacokinetic processes. But here, talk about active secretion, that this step is mediated by transporters. I think I'm going to show you an example on the next slide as to the relevance of transporters. No, not yet, but we'll eventually get to there. So this slide really talks about different factors that can influence drug excretion. So we talk about molecular size and weight at the beginning, and talk about drug concentrations. In urine pH, again, we talk about that. Plasma protein binding, I make a comment that if a drug is bound to plasma protein, then it cannot undergo other process, including drug excretion. It will stay in the circulation. Only when it becomes a free molecule, then it can be filtered through glomerular capillaries into the filtrate. Now, renal blood flow. So again, this can influence glomerular filtration, and therefore influence the excretion of the drug. Now, impaired renal functions. So I briefly touched on the issue of aging and disease, but sometimes the use of other drugs can also influence renal function. One typical example is the use of non-steroidal anti-inflammatory drugs. What happens is that it can lead to acute renal dysfunction, because what happens is that it inhibits the production of a molecule, prostaglandin E2, that normally causes the relaxation of renal blood vessels. And by inhibiting the relaxation of those blood vessels, the effect is essentially the constriction of those blood vessels, and they will restrict the blood flow and therefore lead to impaired renal functions. And lastly, transporters. You see whole range of transporters expressed on both sides of proximal tubular cell. Just bear in mind that when we talk about active secretions, I mentioned that it occurs mainly at the proximal tubules, and that process is mediated by transporters. Now I hope it's very last slide for this lecture. So again, going back to drug-drug interactions. Now I'm going to use an example here, cephalacin. So it's an antibacterial drug, belong to the cephalosporin class. And probenecid is actually another example of a pharmacokinetic enhancer. Now you will see here again the plasma concentration of cephalacin in the presence and absence of probenecid. So probenecid is an organic anion transporter inhibitor, which normally is responsible for the excretion, active secretion of cephalacin. So when you inhibit this particular transporter, you will see that the plasma concentration of cephalacin increases. That's because transporter-mediated active secretion is inhibited, and therefore the drug stays in the systemic circulation longer, and therefore the concentration is higher. Now that shows example of potentially the clinical application of a pharmacokinetic enhancer by inhibiting the transporter that normally is responsible for the active secretion of a different drug. Now actually talking about probenecid, when the penicillin was first introduced, the availability is limited. Probenecid was actually used to inhibit the renal excretion of penicillin so that it stays in our body longer, because if it stays longer, then you don't have to use that much, and then you can use for a wider population. But now, these days, we can produce penicillin easily, so it's not used anymore. As in, probenecid is not used in that context as a pharmacokinetic enhancer. But scientists have looked at the potential clinical application of probenecid in combination with other drugs so that probenecid can be used as a pharmacokinetic enhancer. Yeah, there were some studies recently to look at this thing. Now, just quickly touch on the nephrotoxicity. So I said that the kidneys are an important organ for drug excretion, and therefore the organ is susceptible to drug-induced toxicity. So I am not asking you to remember any of those potential ways by which a drug causes toxicity. I do touch on non-steroidal anti-inflammatory drugs here. If you can also look at aminoglycosides, which is also an antibacterial drug, it can also cause toxicity to the kidneys. But there's some other ways by which drugs can do that. Either they accumulate inside the cells, or they inhibit the transport of other drugs, and so on. But just bear in mind that drugs not only produce therapeutic effects, they can also produce toxicity to different organs. Okay, so I think that's the end of my first lecture. What's the time? Okay, I hope everything is recorded. How do I check? It is? Okay, I should stop talking and take a break. Any questions? I think, yes. Okay, let's take a break. I'll turn off the microphone. Yes. Okay, we are going to continue and we're going to look at the other side of pharmacology, that is pharmacodynamics. Asking a question, what a drug does to a body. Okay, again, you see a list of lecture and outcomes, and I won't go through those in any detail. Now, so you probably remember this slide at the very beginning of my first lecture. So now we are going to complete the other half by looking at drug targets. So drug targets, they refer to proteins or other biomolecules to which a drug binds directly to produce a biological effect. Well, I should point out that the vast majority of drugs do bind to a drug target in order to produce a biological effect, and therefore identifying, understanding these drug targets are important. Now, when we look at drug targets, drugs can bind to proteins or biomolecules, and they can bind to proteins either from pathogen or on the human side, and similarly, the biomolecules can come from either the pathogen or humans. Now, I'm going to use two examples here without going into much detail. First, let's see whether this works. Yay, it works. Can you see the thing? So this shows different antibacterial drugs and how they may work. Different drugs will have different mechanism of action. They can either inhibit cell wall synthesis or protein synthesis, DNA replication and metabolism and so on. But regardless of how each drug works, that you can always identify a target. These targets, in this case, are coming from the pathogen that caused the infection. Similarly, anti-HIV drugs, now you will see the vast majority of these drugs do inhibit a protein from the virus. But the first one, CCR5 and CD4, they are actually found on the host cell in a human. So they illustrate that drugs can target both human or pathogen proteins. And you see these targets are all proteins. But you will find other examples somewhere else where the target is actually a biomolecule. This highlights the importance of proteins, whether it comes from the pathogen or human. They are important drug targets. Now, in the context of this lecture, I'm going to focus on human proteins as drug targets. And later, I'm going to touch on one particular group of human proteins as drug targets and introduce you to some important pharmacological concepts using that particular example. Now, so yes, as I said, that the lecture will focus on human proteins as drug targets. And you see that human proteins are broadly divided into 4 groups: receptors, ion channels, transporters, and enzymes. Here are some examples to illustrate this. Receptors, either they are sitting on the cell membrane of a cell or they are found intercellularly. I think next week my colleague Dr. Liz Johnstone will talk about G-protein coupled receptors example to illustrate drug development. So I think I'll briefly touch on receptor in this lecture so that it gives you an idea of what she's going to talk about. I think it's next week if I'm correct. Now, transporters. While we talk about transporters in my first lecture, so they are responsible for the movement of molecules, endogenous molecules, and also in the case in my last lecture, they're responsible for the movement of drugs. So the second one, ion channels, allows the movement of ions. That can be important for particularly neurotransmission, for example, or many neurological disorders, and so on. The fourth one, kinase. So enzymes, in particular kinase, some kinases are targeted by drugs to treat cancer, to inhibit the signaling pathway induced by these kinases. But let's give you an idea of the 4 groups of proteins as drug targets. Now, in the interest of my lecture, I'm going to focus on receptors because by training I'm a receptor person, then there's no point. Why should I talk about receptors? Because I probably know more about receptors, slightly more than the other three groups, but not much. But I am absolutely fascinated by receptors. That's why you're going to learn more from my colleague next week. Okay, so going back to the 4 groups of proteins, and you will see receptors, ion channels, transporters, and enzymes again. Now, this 2 pie chart shows you the drug targets in each families and the number of targets targeted by a range of small molecule drugs. I want to draw your attention to the pie chart on the right hand side. You will see that quite a significant proportion of drug target G protein coupled receptors, and there's another portion nuclear receptors. So receptors collectively are important drug targets, and that's why next week you learn about GPCRs, because it does represent an important receptors that act as drug targets. But that's the take-home message from this slide, is just to know again the 4 groups of proteins as drug targets, and then also the importance of receptors, which will be the focus of the second part of this lecture. Now, we are going to use receptor as an example to illustrate a few concepts when we look at drug-target interaction. So when I say drug-target interaction, I think to a large extent I'm referring to receptor because it's an example I use here to illustrate important concepts. Now remember, when I say drug targets, it says that molecules that bind directly to the drug target in order to produce a biological effect. Now the reason a drug binds to a drug target, or a drug that binds to a drug target, is because of the existence of affinity, which describes the ability of a drug to bind to its target. The reason then they can bind, obviously because of affinity, but importantly, that's because they are 3-dimensionally fit. So when we talk about a target and the binding, we describe that a target, whether it's receptor or enzyme, has these small cavities, or known as binding sites, that can accommodate a ligand, so a drug for example. And this binding site is the result of a specific arrangement of amino acids, including their functional groups. So the orientation, the charge, and so on, the chemical attraction will determine how well a drug binds to a target. Now, different drugs may bind to the same target, but it depends on the chemical force of attraction. One drug can bind to the same target better than the other, and therefore we say the drug has high affinity than the other drug and bind to the target more strongly. Now the diagram here, so I think one example, so layman language, is look at key and lock. The shape of the key needs to fit with the shape of the lock so that the key can open the lock. But if there's any mismatch, either the key won't be able to open the lock at all, or in the case of a master key, a key can open many locks, which is not desirable at all, because you just want to target particular drug targets to produce a specific biological effect, rather than a drug that binds to a whole range of targets, not only produce therapeutic effects, quite likely it will also produce undesirable effects or toxic effects and so on. So one drug and drug target should be specific as much as possible. This ensures that the drug produces desirable effects but not other undesirable effects. So this just shows that if the shape doesn't match, then the ligand or the drug will not bind to its target. And sometimes this is used to design a drug so that to make use of the space in the target that normally not occupied by the existing drug, But by introducing an extra functional group, for example, to the new drug, then hopefully the 3D fit is even better, so that it then confers selectivity in terms of drug target. Now, talking about affinity, then we are going to look at receptors specifically and look at how drugs may act on a receptor. So I'm going to touch on agonist and antagonist. I'm not going to talk about modulators, although my colleague may touch on that next week, but I don't know. I have not looked at her lecture yet. So drugs that target receptors can be broadly divided into agonist and antagonist. They are both ligands. They both bind to a receptor. And therefore, both drugs, or both agonists and antagonists, have also affinity for their receptor. That's what's common between agonist and antagonist. What is different between agonist and antagonist, though, is that agonist binds to and activates receptors. So they mimic the actions of endogenous ligands. On the other hand, antagonists, they also bind to the receptor, but they do not activate the receptor they bind to. By doing so, they in fact block the action of endogenous ligands. So you can see they are both three-dimensionally fit with the binding site on the receptor, but one is able to initiate signaling pathway, which I'll talk about on the next slide. The other one binds but does not activate the receptor, but both show affinity at the receptor. Now, the other thing when we look at the interaction between a drug and a receptor is look at efficacy. So the ability of a drug to produce a given response from the target. Now keep in mind that when we look at agonists, it activates receptors, and therefore it will allow the initiation of signal transduction, and eventually this will lead to a change in cell function, and therefore we call agonists show efficacy at the receptor. On the other hand, antagonist does not activate the receptor, and therefore it does not allow the initiation of signaling transduction, and therefore you don't see a change in cell function, and therefore we say antagonists do not show efficacy at the receptor. Okay, so these are two differences. Both agonists and antagonists show affinity at the receptor, but only agonists show efficacy. Agonist does not, because it does not activate the receptor. Now, when I say agonists, not all agonists act the same way with the same receptor. Different agonists activate the receptor to different extents, so I won't touch on this too much detail, but there's actually a clinical example when you talk about opioids. Then the treatment for opioid use disorder is to use a partial agonist which is able to produce effects that is similar to full agonist opioids, but it does not produce the same maximum response. So this is particularly useful to avoid potentially the effects associated with overdose. We're now going back to these three things. Full agonist produce maximum effects because it's able to initiate high level of receptor activation. Now, partial agonist, it can still activate the receptor, but it only be able to trigger low level of receptor activation. So therefore, you can still see some effect, but the effect is smaller, and it will never reach the maximum effect that would be induced by a full agonist. And when you talk about antagonist, remember it has no efficacy because it does not activate the receptor. So you see that the effect is just a flat line because itself is unable to activate the receptor. So that's efficacy. It tells you how well a drug is able to produce an effect from their target or their receptor. Now then comes to another important concept, that is potency. So it tells you how much of a drug, so often expressed as the concentration. This is typically the case when you conduct in vitro studies, but when you look at in vivo studies or human clinical trials, then you're talking about dose, because we don't know the concentration in our body, or it cannot be measured easily. We just know, okay, I'm going to give these patients this amount of drug, and then we'll see what happens in terms of the effect. So it tells potency, it tells you how much drug is required to produce a particular level of effect. Now, we can measure the potency for both agonist and antagonist. I'm going to focus on agonist potency here. So it's commonly measured as effective concentration 50%. So it means the concentration required in order to produce the half of the maximum response to the agonist. Let's say there are two agonists, A and B here. So on the left-hand side is a curve showing the relationship between the response and drug dose. And the right-hand side shows the relationship for drug B. Now, so if the maximum response is 100% for both drugs, and then you work out the 50% mark on the y-axis, and you draw a straight line, horizontal line, and intersect with the curve. And from there, you identify the concentration or the dose on the x-axis. That is the agonist potency, or the EC50 for both A and B. Now, in this case, you appreciate that the EC50 value for drug A is smaller than drug B. Let's say drug A is around 10 and drug B is around 100, and therefore drug A is more potent than drug B. So there's inverse relationship between potency and EC50. So for a drug that has high potency, it has a small EC50 value. It's also true the other way around. If a drug has a small EC50 value, that means the drug has higher potency. So that's how you can interpret from these two curves. Now, antagonist potency can also be measured. This is commonly measured as IC50. So the inhibitory concentration of the agonist required to reduce 50% of the response to an agonist. So I didn't show a curve here because for you it's probably sufficient to understand how to measure agonist potency, because that's probably more important when you look at the action of different drugs. But there's a third concept. So we talk about affinity, talks about the binding, and efficacy, we talk about the level of response, and potency, the amount of drug that is required in order to produce a particular response. Now what's next? Okay, now we're going to look at some examples. Well, not specific examples, but just look at the use of drugs to target receptors. I briefly mentioned that when we look at drugs that target receptors, they can either be agonist or antagonist. So we're going to begin by looking at drugs that act as receptor agonist. So it's very straightforward. It binds to the receptor and form a complex. This will often initiate a conformational change that is related to other regions of the receptor so that it can stimulate the signaling pathways, and eventually it causes a change in cell function and produce an effect. It's very straightforward, just like what endogenous ligand in our body does. Now for antagonist, I'll begin with receptor antagonist, and then I'll touch on some other ways by which the agonist-induced effects can be inhibited. Now we can talk about antagonist. Again, it binds to the receptor and form a complex, but this binding does not allow a favorable conformational change that can be related to other regions of the receptor, and therefore it will not lead to the initiation of a signaling pathway, and therefore it will not lead to a change in cell function. Now, that is a very common mechanism, common way to develop drugs to inhibit the activation of a receptor, which can be responsible for many clinical conditions. But that's just one way by which drugs can be used to inhibit agonist-induced receptor-mediated effects. One way is to use receptor antagonists, but there are many other ways to inhibit this whole process. So this gives you an overview of some other ways. First is look at chemical antagonists. I think I will touch on that on the next slide. Or you can use modulators, allosteric modulators, to either affect the binding between agonist and the receptor, or the ability for the complex to initiate a signaling transduction or cause a conformational change. You can also use signaling pathway inhibitors. That is particularly important in treating cancer, using small molecule inhibitors to inhibit kinases that is responsible for the signaling pathway to inhibit signaling pathways in tumor cells. The last is to use functional antagonists to produce effect that is opposite to what this agonist does in order to inhibit the effect. So this gives you an overview of different ways by which the agonist-induced receptor-mediated effect can be inhibited. And we talk about receptor antagonists as one example. And on the next slide, I'm going to take you through two more examples without giving specific drugs. First is, again, chemical antagonists. This refers to drugs that can bind directly to a soluble agonist. So by binding to the agonist, it prevents the binding of the agonist to the receptor, and therefore this way can also inhibit receptor activation. Now, in terms of chemical antagonists, there are two ways. One is monoclonal antibody. So this antibody is against the endogenous ligand, which normally act as an agonist. The other way is to use this soluble receptor, which mimics the endogenous receptor, except that this soluble receptor is unable to initiate signaling pathway. So either way, the endogenous agonist is hijacked by antibody or the soluble receptor, and both of which inhibit the interaction between an agonist and a receptor, and therefore the outcome is to inhibit receptor activation. Okay, this one way is to target the agonist itself, either through an antibody or a receptor. Now, the other example is the use of allosteric modulator. So I'm going to look at MICR5817. Again, this is an anti-HIV drug. So I'm not sure whether you have looked at this, but normally the first step of the HIV life cycle involves the interaction between GP120, which is a glycoprotein on the surface of the HIV viral particle. And eventually it will first bind to CD4 on a CD4 positive T cell, and that interaction will then cause a conformational change to allow the binding of CCR5, which is a co-receptor with GP120. What MICR5817 does is that it binds to a site on the CCR5 receptor to cause a conformational change, so that the binding with GP120 is no longer possible. And by doing so, it inhibits the HIV viral particle to inject genetic material into the host cell. So this interaction is important because it will lead to a formation of channel to allow the RNA genome from the HIV to be injected into the host cell. By inhibiting such interactions, the genetic material is unable to be injected, and therefore it prevents the T cell from being infected by HIV, therefore no further HIV infection. So this example of how an allosteric modulator can be used to change, in this case, the affinity of the agonist, which essentially is GP120, for the receptor, in this case, is CCR5, in order to, not necessarily to inhibit the signaling transduction, but inhibit the interaction between HIV and the host cell. Now, just I mentioned that monoclonal antibody as a way to bind to endogenous agonist, so as to inhibit agonist receptor binding and therefore receptor activation. I'll just briefly touch on the importance of monoclonal antibodies as drugs. Now, the first drug was approved, first antibody was approved not that long ago, 30 years ago, and took another 30 years also to get to 50. But then from 50 to 100 antibodies only took 6 years or so. So you can see how rapidly things are developing as our understanding and technology improves. And it's primarily used to treat cancer and some inflammatory disease, but you can see in terms of the sale, we're talking about in the order of $1 billion. So if you want to make big money, go for antibodies, not small molecules, because the reason is that you can have many generic brands once the patent expires. You can just, make small molecules easily. You just use different formulations, but antibodies, you can't just, make another drug look similar. It's quite highly specific, and it's expensive to develop, and that's what they sell for big price. I can't remember the most expensive drug. I can't remember whether it's an antibody. I think it's for, I can't remember, but I think it's $3.5 million. One dose, I think. So imagine if you find a drug like this, you sell a few doses, well, you'll be a billionaire in no time. So that's just like for your information in terms of the development of monoclonal antibodies as drugs. Remember, they are big molecules, they are biologics rather than small molecules. Okay, now we talk about affinity, efficacy, potency. One last thing, we look at selectivity. Selectivity is important because we want the drug to target the target we intend, not other targets, because it will cause a range of effects that are often undesirable. Now, why drugs show selectivity is because they have different affinities for different drug targets. When we say selectivity, it's only a relative concept, because if we increase the concentration of drug, eventually this selectivity will be lost. So just keep in mind, it's not an absolute selectivity. It's a relativity. It's dependent on the concentration of the drug concerned. Now, why is it important? So this simple diagram shows what may happen with a non-selective drug. So it binds to two different targets with similar affinity. So one target will produce therapeutic effects, but the other one will produce adverse effects. So that's not desirable. That's because the drug does not show a strong selectivity. But on the other hand, if you have a selective drug that binds with the target with high affinity, and this will produce therapeutic effects, that's what we want. And because it has a low affinity for the other target, that means that the adverse effect is minimized or not at all. So this is our goal, is to produce a drug that is highly selective for the target we want to target, but not some other drug targets or receptors that are not relevant to the therapeutic effects. So that is the goal in drug design and development, is to be as selective as possible. Otherwise, you will end up with all sorts of adverse effects. It may outweigh the therapeutic effects the drug produces, which would be not good at all. Now, okay, this is the end of pharmacodynamics, or pretty much, because the next part I will go back to pharmacokinetics and then look at two important curves before I wrap up my lecture on PK/PD consideration briefly on the use of antimicrobial drugs. How are we going so far? Oh, good? Okay. PK. So I mentioned plasma concentration time curve a couple of times in my first lecture, although I did not elaborate on that as to what it means. Now here we come. I'm going to take you through the importance of a plasma concentration time curve. Now again, I am talking about this curve in the context of orally administered drug, because the curve will look different depending on how the drug is administered. Now what we learn, what is important from this curve is that it will determine the therapeutic window of a drug. Why is it important? Because if the plasma concentration of a drug is below the lower boundary of the therapeutic window of the drug, then it will not produce therapeutic effects. If the plasma concentration of the drug is above the upper boundary of the therapeutic window, then it will not only produce therapeutic effects, but it will also cause toxicity. So the ideal situation is that to maintain the plasma concentration of a drug to be within therapeutic window. Now what I show here is one single dose, okay? But quite often a drug needs to be administered at different frequencies, because you see that eventually the plasma concentration will drop below the minimum effective concentration, and it is time to add another dose again, so that the plasma concentration ideally to maintain within the therapeutic window of the drug. And that's why drug need to be administered multiple doses at a set interval, is to achieve that. Not too high, not too low, but just about right. Now from here, this plasma concentration time curve will also give you some other important parameters. First is Cmax, so the peak plasma concentration a drug can achieve. And the time point by which this peak plasma concentration is achieved is known as Tmax. Okay, Cmax and Tmax. So in terms of the boundary of the therapeutic window of a drug, we have upper boundary that is defined as minimum toxic concentration, and lower boundary, minimum effective concentration. And from there, we look at duration of action. So this is the time where the plasma concentration of the drug is at least above the minimum effective concentration, okay? And that will tell how long the drug can produce the therapeutic effects, because once the concentration drops below that, then there will be no therapeutic effects. And we talk about therapeutic window, essentially is defined by the boundaries of minimum toxic concentration and minimum effective concentration. Now, the last thing, area under the curve, is represented by the yellow shaded area here. So it gives you an idea of the total exposure of the drug, the body to the drug. This is dependent on both the concentration of the drug as well as the time of the drug. So it's a combination of concentration and time. Now why I am going to talk about this, I'm going to review that in not the next slide, but the one after. So this is an important curve associated with the pharmacokinetics of a drug. Now in terms of pharmacodynamics, we are looking at the concentration of dose response relationship that describes essentially the relationship between the response or the effect of a drug versus the concentration or the dose of the drug. Now you will see the curve here. It's not all or nothing response. It's graded response. As you increase the concentration or the dose of the drug, the effect will increase until it reaches a point where the maximum effect is achieved. Now from there, it doesn't matter how much you increase the dose, the effect will not change or increase further, and therefore it's pointless to continue to increase the dose if the maximum effect is already achieved. Now when we talk about the concentration or dose response relationship, it's not only applicable to the therapeutic effects of the drug, is also relevant to the toxic effects or the adverse effects of the drug. So when we look at pharmacology, but as well as toxicology. So you appreciate that when the dose is too low, then we know that it's probably not going to produce therapeutic effects. The dose has to be right so that it produces sufficient therapeutic effects, but at the same time, it produces minimal or no adverse effects at all. But if the dose is too high, then again, it produce therapeutic effects, but there's a potential for the drug to produce adverse effects as well. So the dose needs to be optimized. Now, why we'll look at we'll look at curves, one for PK and one for PD. What we're going to look at next, again on PD, is that now going back to the curves, one for therapeutic effects and the other one for toxic effects. What we want to do when we extract information from these two curves, particularly in the case of a population study, is to work out ED50 and TD50, so the effective dose that produce a response in the test population, or in 50% of the test population, and toxic dose, a toxic dose that produce a toxic response in 50% of the population. From there, that we can use the information to inform the therapeutic index of the drug. Why do we care about therapeutic index of a drug? Because it will inform how safe the drug will be in the event of a drug overdose. Now, if you look at this example, there's an overlapping where the drug has a narrow therapeutic index. There's a significant overlapping between the two curves. So what it means is that if someone takes too much of the drug, then quite likely the drug will produce toxic or adverse effects. That's what happens with a drug with a very narrow therapeutic index. Now, an example which I like to use again is paracetamol. Paracetamol has a very narrow therapeutic index. That's why paracetamol overdose is common and can be lethal because it causes severe damage to the liver. If you look at that, probably it will not be approved at all because of narrow therapeutic index. On the other hand, when you have a drug with a wide therapeutic index, then it's relatively safe even in the case of overdose because it has not got into the territory of toxic adverse effects. So, and that's what we aim to achieve when we try to develop drugs these days, is to have a wide therapeutic index and also have a wide therapeutic window so that it's unlikely that the drug will produce adverse effects even in the case of overdose. Okay, that leads to the very last slide, is to combine PK and PD. Now you have learned about this plasma concentration time curve that is associated with the pharmacokinetics of the drug, and you also learn about the concentration response curve that is concerning pharmacodynamics of the drug. Now, in the case of antimicrobial drugs, I think this is important. Have you learned about minimum inhibitory concentration? Right? Okay. So depending on the drug, you may either to consider how much time the plasma concentration is above MIC. For some other drugs, it matters what is the maximum concentration of the drug. And for certain drugs, you need to look at the total exposure of the drug rather than just the time above the MIC. So these are some of the things that you may need to consider in the context of antimicrobial drugs. I won't go into any details in the interest of the time, but there's something amongst a whole range of issues that you need to take into account. Not necessarily in the development of drugs, but when you actually administer these drugs to patients, you have to consider a whole suite of parameters in order to optimize the dose, because you want to provide the best treatment and at the same time avoid toxicities. And lastly, you also want to suppress drug resistance, because drug resistance is a major challenge when we look at the development of the use of existing antimicrobial drugs as well as the development of potential new drugs is to consider how likely the drug, the microorganisms develop its resistance to existing drugs or the new drugs that are under consideration. Okay, that is the end of my second lecture. I survived without going over too much time. Any questions on this lecture? Oh, good. Okay, great. Wow, do let Chris and Lena know or me know if you got any feedback in terms of the content.